Good morning, everyone. So today's talk is about how to improve column throughput while maintaining low pressure drops as you scale up your chromatography. So it's a very vexing problem because many of us all through the industry do process development on a small scale, little high trap columns or other columns like that sometimes. And everything seems to go well. And we are simply magically scaling these columns up to, 10 centimeters, 20 or 30 centimeters, then eventually to 1 meter columns. And very often, unbeknownst to the people that develop these separations, they encounter significant problems when these separations are scaled up to pilot or large scale. And that is the worst time to be encountering problems, whether it's with column packing, whether it's with high back pressures, then the skids are not able to handle it, etc. So many people, these problems are not new. Many people are familiar with it. And then it hits the timeline, hits the productivity and throughput gains from that particular plant. So we are going to address those problems. But before we do that, I'll take just a minute to introduce the company, Sepragen, to some of you who may not be familiar with us. So Sepragen is a company that has been in business for over 30 years. And we have over 30 FDA-approved drugs that use our equipment in manufacturing commercially approved biologics. We have over 200 global installations and two major COVID-19 vaccines are made using our equipment. So these are not experimental drugs. These are literally in the arms of thousands of patients. Okay. So, and then of course, Sepragen's COVID vaccine products are well known in the industry, certainly by companies who made their vaccines using our products, and we were able to deliver this equipment in record time, okay, during the Delta wave, for instance. Now, Sepragen is an innovation leader in reducing time to market and reducing capital cost and cost per dose. And how do we do it? We have a number of unique products that we will not go into today, because today's topic is on how to improve column chromatography throughput without increasing back pressure. But just by way of background, Sepragen makes radial flow chromatography columns. These were invented by Sepragen and now used widely through the industry. Lots of experience in the industry as well as the FDA has with these products. A whole line of chromatography systems from the bench to large scale, probably rivaling the leader in this field. So well established. We also have as many as 4 single-use chromatography bench scale and large scale skids. We've got continuous 2 and 3 column continuous chromatography systems, both at the bench scale and large scale. So very unique in that respect. Again, we are all about scalability. And then we have buffer making. You can't really separate chromatography from buffers. So how do you make these buffers more efficiently? So we have a number of products that address that issue. And then how do you integrate this buffer making capability into your chromatography systems, whether they are single use, multi-use, or continuous systems? So we have some products that do that as well. So that was a quick primer. And ultimately, we are all about making your processing installations more efficient. Okay, and Sepragen's products are all focused on bottlenecking your downstream processing. So whether the column is the issue and the pressure drops and throughputs related to columns, whether it's buffer making, whether it's column packing. If you look at this facility right in front of you, a plan of a facility, at the bottom you see the cell culture corridor all the way from the seed train to large scale. And then on the top of your screen, as you round the corner and come to the other corridor, you go through the DSP suites and then to the buffer making and the column packing operations. Now, as the titers go up, guess what? The same seed train, the same bioreactor, bottom side of your screen is going to work fine. But if you make more product, you can make more product in the same bioreactor, but to absorb that product, you're going to need bigger columns. So often that's where the trade-off comes. That's where the pressure comes. If there isn't enough space in those DSP suites, guess what? You're using the same size skids, the same size columns. You're having to do more cycles. So your cycle time's increasing. So your overall productivity of the plant goes down. No, so instead of making maybe 20 batches a year, you're now making 15 or 10. Well, your capital cost is now being amortized over half the number of batches. So your cost of goods is going to be impacted. Or you're going to put additional people if you're doing round the clock shifts, and your variable costs, your FTEs are going to go up, right? You're going to have workflow issues. And of course, as you have bigger columns, well, guess what? You're going to need bigger space to pack those columns. You're going to need bigger kitchens to make the buffers in and more torts lining up that corridor. So that's a frequent problem. And of course, if your columns are flowing slower because you're having back pressure issues, it's like the body having high blood pressure. Guess what? Everything slows down. You can't run as fast. You can't do things as fast. So, the column's kind of the heart of the process here, or at least the downstream part of the process. And if you're not able to get as much productivity and flow out of it, it impacts the productivity of the whole plant. Okay, so that's why we are addressing this issue now. And we come from a base of experience because while many of you may not be familiar with radial flow chromatography that I'm going to talk about now and how it addresses this issue, many of you are. And this technology is proven out there. It's not, for some of you, it may be a new concept, but many others have tried it, and there are a whole host of drugs using these. And so people at these companies, many companies have many lines using this technology. And certainly the FDA is familiar with it. But we'll go through it in a stepwise manner today. And so first, let's kind of define the problem. Chromatography problems, throughput and pressure. What can radial flow, superflow columns do? Well, on the left-hand side is a conventional column that many of you are used to. No big deal, small column, pack it with resin, push the adjuster down, and you pretty much run, 100, 300 centimeters per hour, few minutes residence time, 2 minutes, 3 minutes, 4 minutes, 5 minutes residence time. Everything looks good. Well, then you scale it up, and eventually the sizes that are on the right-hand side Well, certainly doesn't look that easy as the one on the left. You can't just move the adjusters up and down. You've got to have hydraulics moving these things. You cannot just have a 10 centimeter or 3.5 centimeter bed and run a quick um, separation on our bench scale systems. You've got to have large packings. You've got to have large systems to pack these columns. You have to have large systems to run them. If you have very tiny beds or short bed heights, well, the packing efficiency in these large columns is not so great. So they tell you, well, you should have at least a 20 centimeter bed column. Well, really? Because you can do separations, whether it's ion exchange or affinity, even on membranes. You don't need 20 centimeters. Where did that come from? Well, a lot of times because that's the minimum bed height that large columns need in order to pack them properly. Residence time, yes, but that's another matter, and we'll address that shortly. So when you have to change these bed heights as you scale up to ensure that uh, you get even flow through these columns, that they're packed properly, guess what? The higher bed heights give you higher back pressures. And your low pressure skids can only tolerate certain back pressures. And with certain resins, sometimes during loading, sometimes during elution, the back pressures go up. And then again, you have to slow the flow rates down. So that's annoying. It also leads to increased cycle times, increased um, pressures, longer, um, longer times for people, maybe multiple shifts, disrupts workflow, and has certain deviations be recorded in your process logs, and eventually leads to lower productivity. So enter radial flow chromatography. So some of you who are not familiar with it might be asking, what is radial flow? Well, everyone knows what conventional flow is, conventional chromatography or axial chromatography is. Doesn't really need a lot of explanation. You have an adjuster, the flow comes in through the top. There's a frit on the top of this column, at the bottom of this adjuster. And there's a frit on the bottom, and the resin, this white stuff, is sandwiched between these two frits. That's it. So the flow takes place axially along the axis of the column. Well, radial flow kind of switches that paradigm, 90 degrees. And how do we do that? Well, in these columns, the flow comes in through the inlet, in which the radial channels that distribute the flow, not disperse, but distribute the flow to the entire periphery of the column. So inside the column, but the periphery of the column. And so inside the outer wall of the column is a porous frit. So instead of there being a circular frit of the column on the top, there's a cylindrical frit on the outside. There's a cylindrical frit on the inside of the column, inside which is a solid core. So now the resin is held between these two inner and outer frits. So what you do in this column is essentially direct the flow. Instead of being distributed on the top of the column and going through the resin bed, coming out the bottom, here the flow is distributed to the entire outside wall of the column. And then your sample travels through the outer frit, through the resin bed, through the inner frit. And of course, inside the inner frit is a solid core or hollow cylinder. And so between the wall of the inner frit and this hollow cylinder is a narrow channel, and it comes out the bottom. So the area over which now the flow is applied is not the pi r squared, but it is the area of the wall, which is the 2 pi RH. And the resin bed height, is instead of being the vertical bed height, is the annular distance between these two frits, because the flow is going in this direction. Shown better, just as well, on this other diagram. You can see the flow coming in here, going to the outside, going through the resin bed, and coming out the bottom. Okay. So in our larger columns, we don't actually have channels. We have two impingement plates. We have an impingement plate that's suspended below the entry, which disperses the flow to the outside. Okay. So it's like a radial cartridge in which the bed is held. And this gives you a very large area and a very short bed height. So that gives you low back pressures, which is what you want for your bind and elute chromatography, right? For ion exchange, for affinity, and for Hick. And you can get very high throughputs because the area is much more, as you can see from the next slide. So this area of this same cylinder, 50 mil cylinder, is 80 square centimeters. If you look at the annular area, and of this cylinder, 50 mil cylinder, if you just look at the pi r squared, it's 10 centimeters. We have 8 times the area. Therefore, for the same linear velocity, you get much higher flow rates. Because what are flow rates after all? Linear velocity multiplied by area. Okay, in this case, the area is the outer wall of the column. And therefore, you can get very high throughputs in the same, in a column which is taller and narrower, and holds very small footprint. So remember we talked about the titers going up in cell culture and how do you scale up? Well, here's a picture. This is a packing skid that we are showing you here for a column. But here's the column itself. That's a 100-liter column, which is the equivalent of a 1.4 meter column. Okay, so 1.4, 1.6 meter column, depending on what the bed heights are. Well, you got that in a narrow column like this, which is the equivalent of, maybe a 45 or 60 centimeter normal column. Okay, so, and you get that kind of, so not only do you get that capacity of resin in the column, but for that same capacity, you get much higher throughputs. by simply changing the column format. Pretty good news. And that's not the only benefit. In manufacturing is huge. Now, what about for clinicals, you might say? In R&D, yeah, you could cut down the time. What about clinicals? Well, you're not going to really do a lot of runs in clinicals. I might do, I don't know, 8 to, 10 campaigns. Then we'll put our column away. And yeah, it takes me a day, but it's no big deal. I don't see the need to change the column. Well, here's a case study. In this particular case, they had a 250-liter bioreactor, 4 grams per liter of titer, so roughly a kilogram a run. And they had down here 40-liter conventional column packed with protein A. Okay, so about $800,000 worth of protein A in this column. And the CapEx along with the skid and the column, somewhere around $350,000, $400,000. So that's the investment in that step. And the output is that they can do the entire batch in a day. Fine. What's the big deal? Well, the big deal is that's over $1 million for 8 or 10 campaigns, after which all that expensive protein A is just going to sit there in the cold room. Because who knows when the clinical results are going to come in and what you're going to do with that material. Okay, so this other customer looked at it and said, well, if I can go 4 times with faster than with a radial flow column, why do I need to use a 40-liter column? Why not use a 10-liter column? And in the same amount of time, if each cycle, let's say I'm generous, instead of 1 hour, I give 2 hours for the cycle, between cleaning and changeover and all that. I program my equipment to do 4 cycles. I can just use a 10-liter column, do 4 cycles in a day, 2 hours each, and 8 hours, I'm done with the entire batch. And why would I do that? Well, the amount of protein A that I require now goes down from 800,000 to 200,000. The reason is 600,000. And then I have a smaller column and perhaps a smaller skid from another vendor. And now, instead of spending, almost $1.2 million, you spend about $400,000 and you're able to use that for the entire clinical production. You saved on $600,000 worth of protein A. Is that worth it? Some people might say yes. Because after that, even if you did 4 cycles, you're not using that resin to capacity and to exhaustion anyways over 10 campaigns. So if you did 10 runs or you did 40 runs or you did 80 runs, you're still within the life of the resin. But you're utilizing it more fully, and therefore you're some more prudent use of capital and resources than otherwise. So radial flow columns have a benefit even at the pilot scale. Because of their high throughput, you can get throughput, you can have safe time, lower back pressures, and certainly even save expensive resin. Now you might say, well, you're going faster through the column. I'm concerned though. I'm concerned that you're going fast through the column, and what is it really doing for the binding capacity? Am I going to have the same recovery? Well, we've already shown you from, for instance, the Genentech slide that the recovery, purity and impurity profile was all consistent. But here's the theoretical basis for that. So if you look at the dynamic binding capacity of modern day resins, This is what the dynamic binding capacity profile looks like for some common resins, okay? In the old days, this curve would be, this inverse curve would be very steep, meaning that if you were running at 10-minute residence time or 8-minute residence time, the capacity would be very high. You come down to 1 or 2 minute residence time, and your capacity would be in the tank, in the toilet, 10%, 15%, like that. Why? Because these beads were cross-linked beads, and the sites were well inside the bead. So it took the large molecules a lot of time to diffuse inside to the sites and then come out. So the mass transfer limitations were very high. That meant that if you went very fast through this column bed, not all the sites would have to be would be available to the protein for binding. But modern ray resins have changed. So they're macroporous, so the pores are much bigger. And then on top of these beads, there are grafted sites. So they're like bushes, right? With ligands on the ends of these bushes that are now in the liquid solution. So the mass transfer limitations have been removed, and those many more sites are accessible right at the surface. And so whether you go fast or slow, it doesn't really matter that much. As you can see, like if you look at this map select extra, whether it's an 8-minute residence time or 4-minute residence time, it doesn't matter. Capacity is unchanged. It goes down about 5%, going down to a 2% to 2-minute residence time. So, the residence time reduction from 5 or 6 minutes to 2, even 1 minute, may be of the order of 5 or 10% reduction in dynamic binding capacity, right? But what you get in return is a 4x improvement in throughput. Now that's a bargain. That's a bargain. Why do I say that? Well, if you could reduce that column because you're going 4 times faster, you can reduce that column from 40 liters to 10 liters because you're going 4 times faster and you can do 4 cycles. Well, even if you've lost like 10% of dynamic binding capacity, guess what? You just use a column that's 10% bigger. So instead of 10 liters, you're using an 11-liter column. You still saved a lot of resin, right? You may not have saved 30 liters, you may save 29 liters. Okay, still a lot of money. So, you can compensate that for that very easily, but what you can't compensate for is if your cycle time for doing multiple batches, because your titers are high and now you're having to do 8 or 10 batches, and that's taking you a week or taking 2 weeks, and you can cut that down to a few days. Well, you gladly do that because now you can have the capacity of your plant available, which you wouldn't have otherwise have available. And then you might say, well, that's great. I can see how you can reduce, you can reduce the back pressure. Sure, it looks like you're able to go much faster. But, this is a problem that we really see at clinical stage. I'm seeing that with our clinical, but isn't it too late to change from axial to radial? Maybe we'll do it on new products that come along. Well, no, it's not too late. That's a fallacy. Okay. People say that about resins because that directly impacts CMC. You write down in your CMC application that you're using this resin and you're using these buffers and you're flowing at this flow rate, etc. But if you were to change a pump, you would do that without blinking an eye, right? Or if you were to change a valve. So this is a hardware. If you were to change a tank, you wouldn't think twice about it. Well, this is a hardware change. And unless specifically written in your CMC application that you are to use an axial column, there's nothing that's precluding you from doing that, to making that switch. So here's a test case that we talk about. Here's an actual case. This is a case study. So this is a customer, Chiron, and at that time today's Novartis, but at that time they were in phase 2 of making a well-known growth factor, which is now approved. So they have 2 of these that are FDA approved. And this process was done, it's a lab scale work, picture or chromatogram of lab scale separation that was done on a copper chelating resin, okay, on axial column. And so that's the load, that's the elution. Well, unfortunately, this process got scaled up into manufacturing when they saw all kinds of pressure issues. And the process was taking, and of course, with chelating resin, long wash cycles, right? Everyone's familiar with those. So the cycle time is very long, can't be done in one shift. And the process is being done across multiple shifts. And during shift changeovers, etc., there are many problems. And that year, they had already lost 3 batches. When the production manager started the ball rolling and said we have to look at a different column so we can all get this done in one shift. And so they tried a radial flow column. Yeah, no question. We were able to help them get the process done faster. And then there was a question of switching. So we went to the CMC guys and asked them, well, can we do the switch? And they said, well, I don't know. We have to look at the CMC application. Because like anything in validation, you've said, this is what you're going to do. That's what you're going to do, right? You first you tell them what you're going to do, and then you do what you told them you're going to do. So we asked them, well, have you specified what kind of column you're going to use? And they said, no, we've specified the resin. We've specified what are the elution, wash, equilibration buffers. We specified the buffers. We have specified the linear velocity during loading, and that needs to be 100 centimeters an hour or 0.16 bed volume per minute or CVs per minute. So we got our answer, and he said, as long as you keep those, we are good. You can use any column. We've not, and of course, all the final criteria, purity and yield and all that has to work, and you need to make sure the columns are well packed, and you can, the HETP, the asymmetry, and all that look good. So, anyways, that's what was done. Eventually, this was scaled up to a 20-liter column. And the client said, okay, we are going to keep the loading flow rate the same, 0.16 bed volume a minute. We going to adhere to, even though they could have gone a little faster, they didn't. But they said, hey, we can crank up the equilibration flow rate. We can crank up the wash flow rate, which is a big deal. We'll crank up the elution flow rate. We crank up the regen flow rate. So here you can see they did that. and the process time was much reduced. They also found, curiously enough, that the capacity utilization in this column was better for them. So we write that down from 5.3 to 6.85 megs per mil is what they got. Don't know why, but anyways, that's a curious fact. So these unique little wedge columns, how do you pack them? Well, first, how do you pack a radial flow column? On the left hand, you know, the strip over here, you see from the top. First, you look at the top diagram, you fill up the column from the bottom with buffer. And as the buffer fills up, air is displaced from the two packing ports that directly access the annular chamber between the two frits. So once the column is primed of air, then make a packing slurry and pump it in through these packing ports. In large columns, they're like 4 packing ports, 90 degrees apart. And the buffer goes, and the packing slurry goes in, and the excess buffer is driven out from the top until the bed packs up, to this point. And then the flow is switched to the bottom of the column, and the bed packs up in the reverse direction from the outer frit to the center. And then suddenly you'll see the pressure spike. Pressure will go up anyways as the bed is as the bed is being created, but then it suddenly spikes, and that's when you stop the column packing. And then you run the column, close the packing and unpacking ports, and run the column run the column in the forward direction and in the reverse direction and stabilize the bed. And when we want to unpack it, then you pump buffer in and open the bottom unpacking ports, and the bed slurries and comes out. You can pump air in to disrupt the bed, it makes it easier, and then the bed slurries and comes out. And how do you know whether your column is well packed? Well, just as you would any other column. So we have automated packing systems as well. And this is just a chromatogram bed integrity check from one of those columns where you can see here the asymmetry is about 1.18 for this particular pack. So anything which is in the normal range of 0.8 to 1.6 or 1.8, whatever your criteria is good enough, and you can check the HETP and asymmetry. And then on the top here, you see how the wedge is packed. You can simply take a syringe, actually, and with a syringe and a pressure gauge here, and this is the packing port of the wedge, you first, fill buffer in here and drive all the air out. Then you make a slurry and you pump it in, and you see initially the pressure is 0. And then as the slurry goes up and the bed is being formed and packed, the pressure goes up. And, when it's fully formed, then you stop packing, close this port, and you can go ahead and simply run the column. Okay? And then when you want to unpack it, then you pump buffer through the top, open the packing port, and the bed slurries and comes out. So very simple. Of course, we have medium size columns, process columns, large production columns. You can't pack those with a syringe. So you got packing systems, you got diaphragm pumps you can use to pack the slurry into the columns. And we have automated systems. So you have automated systems here for, 5, 10, 15, 20, 50-liter columns. If they're using explosion-proof solvents like ethanol or IPA in the slurry, then we have explosion-proof versions of these. And then for very large columns, like the 100-liter columns we showed you, we have large packing systems like this, where you can simply make a method or make a recipe, just like you would a chromatography recipe. Here you have different packing recipes that can be stored and at different set points, back pressure, the column will start the packing, stop the packing, and the column will be automatically primed and then packed and then bed stabilized. And then you got sensors there so you can check the HTTP and asymmetry and tell you whether you passed or failed. And you've got the entire GMP batch record, an electronic record for that packing. Based on the 21 CFR Part 11 compliant software. And of course, these columns now have been scaled up to very large scale. Uh, whether it's the 30 commercially approved biologics, whether it's mAbs or vaccines or, human plasma fractions up to 20,000 liters. And these large columns that you see are superflow radial flow columns. On the left-hand side are 4 columns running in parallel, processing a very large batch. So tens of thousands to hundreds of thousands of liters. On the right here is actually a very interesting industrial installation. And here we have multiple size columns running in parallel. Okay, so you needed, for instance, one is a 350, the other is a 250, so 600 liters of column capacity, no problem at all, right? And some of these are done, we have installations, large installations of radial flow columns where we are isolating nutraceuticals like lactoferrin from milk or skim milk, running 1 million liters a day. So here's an example of such a process, excuse me, where they are running huge amounts of whey through a radial flow skid. And on the left-hand side is where this process was being developed on one of our smaller pilot units in our food plant. Excuse me. And what you see on the right-hand side, of course, the salmon-colored liquid is the lactoferrin. So that's the output. So that gives you a sense of how you can go at such high throughputs. So whether it's tens of thousands of liters, 1 million liters a day, very high throughputs with no back pressures. It's done, it's been done, and these installations are operational for decades now, so it's proven. And if it's something that's you guys are new to, certainly encourage you to reach out to us. We'd be happy to work with you and show you how you can do this rapid scale up from the bench all the way to large scale without encountering these issues. And you can work in very small footprints and get very high throughputs. Now, and of course, these are very large installations. And just a little bit, so these columns, you can use anyone's skids with these columns, anyone's systems or skids. But if you find you're limited in terms of the ability of those skids to run at higher throughputs, we certainly make Chrome systems as well. Right, so all the way from the bench scale to on the right are 30 liters a minute, 60 liters a minute skids, and they come with all the sensors, all the valving, UV, pH, conductivity, gradient functions, 21 CFR Part 11 compliant software. You don't need these to run our radial columns. You can run them with anyone's skids, but should you decide to look at these skids, we can certainly provide them for both our radial flow columns or conventional columns. Lots of benefits for these things that I won't talk about today. And then if you're using pre-packed columns or interested in pre-packed radial flow columns or not, you may be using other people's columns. We also have a line of single use with chromatography systems, meaning that the entire flow path, including all the sensors and all that, are disposable, all come pre-assembled, pre-sterilized, if that's something of, and again, very easy to use software, very easy to change the flow kits, and very low hold up volume, low footprints. Happy to talk about you if there's any one of you in this group or people that you might know that are interested. And then we're talking about high throughput, high productivity. You can use radial flow columns, but uh, many people are going to continuous processing. So again, in continuous, whether it's two column, three column, or four column processing, if you're having the flow of one column, the flow through from one column going to the next, the radial flow columns are very good. They can be used to give you the high throughputs without the back pressures. They're ideal for that. And then we also make these continuous chromatography systems, whether on the right-hand side, you see a three-column bench scale continuous system, and on the left-hand side, you see a 600 LPH continuous three-column chrome system, complete with software and everything that we provided for large-scale manufacturing. And these come incidentally with the innate ability to create their own buffers. So you don't have to, if you're doing continuous production, you don't have to make huge amounts of buffer because you can just use Wi-Fi and the stock acid and base component of the buffer, concentrated acid and base components, and then the salt, and all of them will blend in together to give you a 1x buffer, whether it's an equal or wash solution buffer with the right pH and conductivity. So this kind of P&ID diagram shows a running screen of um, one of those runs. And then we do a lot of customized um, project management. So everything from custom skid development to customized columns. And we work with you on a project basis. And we have, again, mechanical, electrical, software team. We do a lot of our own manufacturing and assembly. So we can certainly work with you on timelines. There were some products during the COVID era where we helped the company scale up their vaccine production from 40 million doses a month to 200 million doses a month. And we did that in 18 weeks in one case and 13 weeks in another case. So we have a lot of our own internal in-house capability that enables us this level of flexibility, nimbleness, and speed. And then we provide you with full-blown GMP support, whether it's FAT, SAT, IOQ, a complete doc package, all the GMP test certificates and all that you guys might need. And our goal eventually is just to help you debottleneck your process, speed it up, and whatever is blocking you, whether it's pressure issues or footprint issues in your plant that reduce the productivity or reduce the workflow, create bottlenecks, those are what we are out to solve, and we are your partner in that. And so that's who we are. We deliver what we promise and would love to work with you on the challenges that you are facing. So with that, I conclude this webinar and am open to any questions that you might have.